Hello, and in this video we are going to talk about the process of mitosis. So mitosis occurs in the cell cycle, and it's just this little part of the cell cycle. It's much shorter than interphase. So we said that an actively dividing mammalian cell interphase is about 12 to 24 hours. So definitely actively dividing cells. The mitosis part will be 1 to 2 hours. And what we do in mitosis is we make sure that all the energy is into dividing the DNA safely and forming two cells. So the cell will stop all of our other activities. So we don't do protein synthesis. You do all that beforehand and you just divide that DNA and divide that cell safely. Mitosis is used for growth, repair, and asexual reproduction. And kind of the important terminology we are looking at, we have a little picture like this. What we're going from is the original cell that we call the mother cell, and we produce two daughter cells. We should put a nucleus in there as well. So what we have here are genetic clones. So the daughter cells are genetically identical to each other, but they're also genetically identical to the original mother cell as each other. So we're producing clones. If we just mention asexual reproduction a little bit, so we're going to talk about meiosis to produce gametes like sperm and eggs and do sexual reproduction. Lots of organisms actually reproduce asexually, things like bacteria, also some plants. So here we've got a strawberry plant, and strawberries plants can replicate sexually. They have flowers and all the rest of it, but they also can replicate asexually, and they do this by doing mitosis. So this is the original mother plant, and then send out this here, it's called a runner, and when they send the runner out they get a baby plant, and it is a clone. So this is a clone of the original. So this is the original and this is the new plant, and they are clones of each other, so they can also replicate asexually as well. Mitosis itself, you get the classic picture here where you see all the different phases, and you can look at a phase and work out whether it's in interphase or metaphase and so on. But it's really important to remember that it's actually a continuous process. So a cell doesn't go, I'm in prophase, and then jump into metaphase, and then jump into anaphase. It's a continuous process, and you might look at an image and try to work out, it could be early metaphase, it could be late prophase, for example. So it's definitely a continuous process, and it's not always very easy to say definitively what stage you're in, because in real life the mitosis doesn't always look as beautiful as it does in the books. Well, it looks as beautiful but not as clear cut because it's a continuous process. The first stage of mitosis is prophase, and what we've got here is an image taken by a light microscope and a cartoon. Now the thing to notice with the light microscope picture is all these structures that you talk about and you see in textbooks, you can't see in the light microscope. What you can see in the light microscope are the chromosomes. So they're the things that look like purple worms in the nucleus. So what's going to happen in prophase is that the chromatin will condense to chromosomes. So you'll see the classic chromosome structure like this. And so the DNA is kept into short condensed structures that are easily divided, and they're held together at the centromere to make sure we're going to get proper division with each daughter cell. We're getting one copy of the DNA. So each arm is made up of a sister chromatid. In prophase as well, the nucleolus will disappear. Remember, the nucleolus is involved in ribosome production. We're not going to be producing ribosomes anymore. We are focusing all of our energy on getting this DNA divided safely. Microtubules, so a type of protein called microtubules, will form the spindle fibers. And these spindle fibers, I'll just make that yellow. These spindle fibers are shown on the cartoon like this. You can't see them on the light microscope picture, but these spindle fibers are the structures that will pull the chromosomes and move the chromosomes around. The other thing that will happen is that the centrioles in animal cells, which are involved in cell division, they will move to opposite sides of the cell. And these centrioles, which I'll highlight in red, this again, you can't see them on the light microscope picture, but these centrioles are the centers that will organize where the new cells will form. Towards the end of prophase, you will have a stage called prometaphase. You can't tell it as looking any different to prophase with a light microscope. But what's happening in prometaphase is the nuclear envelope will break down. So that nuclear envelope is there to keep the DNA safe away from the rest of the cell, and you've got that very tightly controlled entry and exit to the nucleus. We are going to break this nucleus and split it into two and form two new cells, so we need to destroy the nuclear envelope, so we can do that. And the chromosomes will each bind via their centromere to the spindle fiber. And they will be therefore ready to be organized and moved around the cell. Metaphase is often the easiest cell stage to spot when you're looking down a microscope. And in this stage, the chromosomes move to the center of the cell. And this center gets called the metaphase plate. So the metaphase plate looks like this in the light microscope picture. So with this lining up, if I attempt to draw it with my amazing drawing skills, so we've got, this organism has 3 chromosomes, like this, and they've all got their lovely centromeres. We've got the centrioles on either side, and then we've got the spindle fibers like this. So notice each chromosome is attached to a different spindle fiber, and they are lining up in the middle of the cell. This will be one cell, this will be the second cell, and they are lining up so that we will get one sister chromatid going one way and the other sister chromatid pulled the other. So it means that when we do start to do the cell division, it makes sure each cell gets one copy. So each daughter cell gets one copy of the DNA. And remember, we've got a checkpoint here. Okay, so when we talked about cell cycle checkpoints, you've got a checkpoint here that will wait until they all line up nicely in the middle. If they don't line up, we'll just stay here and wait until they do. The next stage that occurs is anaphase. Now, we said that the chromosomes line up and they're attached to the spindle fibers via their centromeres. What happens in anaphase is that we are going to split. The centromere is going to divide. So in anaphase, your DNA will split and you get the centromeres divide. So the centromeres divide like that. So the sister chromatids are going to pull apart. as soon as they separate, they're now referred to as daughter chromosomes. So now called chromosomes, or daughter chromosomes if you prefer. So as soon as they've been pulled apart, we call them chromosomes. So this, I'll do that in a different color, you can see it, this is now a chromosome. When they were together, they were called sister chromatids. As soon as you pull them apart, they're now called chromosomes in themselves. You get this classic V shape when the cells are in anaphase. So if we've got the cell like this, and I'm just, for the ease of my drawing skills, we're just going to have one chromosome here. We're pulling by the spindle fibers like this. So we're pulling to the centrioles like this, and what you find is that the arms of the chromosomes stream behind. And the way to think about it is the centrioles are pulling on the spindle fibers, and they're pulling on the centromeres, and the arms of the chromosome are streaming behind as they are dragged through the liquid, the cytosol of the cell. So the arms drag behind. So you see this classic V structure like this. Then we have telophase. Now in telophase, what happens is the chromosomes, as they're now called, are at opposite sides of the cell. So you can see opposite sides in the light microscope picture. The spindle fibers have finished their job now. So spindle fibers disappear. We don't need them anymore. We want to keep our DNA safe, so new nuclear membrane is formed. So that forms, there's two of them around each nucleus in the two different cells, and the chromosomes will relax or decondense back to chromatin, and in this relaxed form we can now restart protein synthesis, for example, and transcription. And you'll no longer see the individual chromosomes under a light microscope, you'll just see the chromatin. So that's what's happening in telophase. We then have cytokinesis. So cyto means split, kinesis is cyto is cell, kinesis is split. It occurs slightly different between animal and plant cells. It's enough at this level just to say the cell splits, but we can mention it to begin with. So what happens in animal cells is what we call a cleavage furrow forms. We get the proteins that make up the spindle fibers all kind of make this smaller area, and that pulls the membrane in. And then when the membrane is pulled in, eventually it's pulled so close that they fuse, and you end up with two cells like that. Bit more complicated with plant cells because they have the cell wall. So you can't get this kind of furrow forming and this pinching forming. So instead what happens is vesicles from the Golgi apparatus form in the middle at the equator. So they move to the middle of the cell and they fuse with each other. So you end up with like fused vesicles in position, and this, as the vesicles fuse, they actually separate the cell membrane. So the cell membrane is separated, and then the plant can lay down new cell wall to separate the cells properly. So that's what happens in plant cells, but you don't need to know the details or remember the details of cytokinesis at this level. So just finally to look at some beautiful pictures of images. So this is the image we saw before, and we mentioned on this image previously that lots of cells are in interface in here. But we've got the other stages occurring too. So this stage here, hopefully you can work out this one is prophase. We have got a beautiful metaphase here. It's always a thrill to find a metaphase. Here, another metaphase as well. Maybe a slightly earlier metaphase than this one, because you can see the chromosomes are not lined up as neatly in the middle. And here, hopefully you can work out we've got an anaphase. And I think maybe, it's quite hard to tell, but I think maybe this is actually just at the end of kind of cytokinesis, because these two cells look like they were once, and they're starting to fuse. Starting to, not fuse, break apart into two cells. So starting to do cytokinesis. So that's with a light microscope. This image is taken by my favorite. This is taken by a confocal microscope. And what we have in this image are three cells. We have got, this cell is prophase, and we have a metaphase, and we have, this is an anaphase. You've also got lots of cells that are in interphase in here as well. Now, it's a confocal microscope. It uses fluorescent proteins, fluorescent tagged proteins, and laser beams. And we've got three different colours on here. So if I colour code it, the yellow colour, which doesn't write very nicely on my screen, those are the centrioles. So these structures here are the centrioles. So hopefully you can spot the centrioles on the picture. Just underline that so you can actually read it. The red structures are the spindle fibers, so the microtubules, which doesn't want to write, which is weird. So spindle fibers, like that. And the blue, finally, is the DNA. And what you can actually do with a confocal microscope is you can watch mitosis in real time in living cells, which is always thrilling. Thank you.